introduce Jim Grace. Jim went to college at Presbyterian in South Carolina, graduated in 1973, did a master's at Clemson, and then a PhD at Michigan State University in College Station. He finished in 1980. He went on to be a faculty member at the University of Arkansas, and then at Louisiana State University before he joined USGS, which is where he is now. He's won lots of awards, but the most recent one, he was elected a Fellow of the Ecological Society of America in 2014. So if you look at the list of fellows, it's a pretty impressive list. He has published over 170 papers and reports, including 3 books. So that's the stuff that you will see if you go to the website. I've had the privilege of working with Jim over the last few years on some papers, and so I got a little bit more of a first-hand introduction. And I think what really made an impression on me about Jim is that he has a really deep interest in the philosophy of science and what you might call the science of science. Why is it that the scientific method works? When does it work well? When does it fall apart? And I think his interest in that sort of meta topic drives the kind of work and kinds of approaches he adopts in his on-the-ground research. That led him away from traditional statistics to structural equation models, which we'll talk about today, I think, which has become a major focus of his work, teaching people like us how to use those approaches. He thinks very deeply about how theory is derived, how it's represented in models, and tested with statistics. And he cares a lot about getting it right. I think more than we're all supposed to care about getting it right. Sometimes we end up caring more about getting it published. And I think, compared to Jim really sets a nice example in terms of working very hard to make sure we feel good. Join me, welcome.
Well, first, thank you, Peter, for that introduction. I really appreciate it. And thanks also to the grad students, the ecology grad students, for the invitation and a chance to come back. It's been a few years since I was here, but not too many, and it's always delightful to come back to this part of the country and to Utah State. So, preview at the end of the talk. That's a good way to start. What do you think? So what I want to do today is to talk about a debate for the purpose of revealing some issues about how science progresses and sometimes doesn't progress in the ecological sciences, and also to talk a bit about history and some of the trends that have moved us forward or failed to move us forward as well, and focusing on this productivity diversity debate, which on the one hand may seem quite abstract, but I think that there's a fascination. I don't know if it's yet gotten to be a morbid fascination, but I think everybody out there who sees this has to be fascinated with the topic that we have worked on for so long, and there's so many papers, and there's so many experiments, and it seems that the debate just amplifies and forks, and resolution becomes very difficult to this topic. And I do have hope, by the way, that we'll get to some resolution or some clarification in part, but the question that we're trying to address is, can we make any progress in sorting out the connections between these two concepts and the measurements of these concepts, diversity and productivity? And there are a whole host of different theories and models that have been presented, but the most conspicuous feature of all this has been the back and forth, no, it's not right, I don't agree with that, your data don't fit, yes, my data do fit. I've just been working on a paper which Peter's an integral part of, and it's my collaboration with him that's really driven this forward. We even have his picture that will show up in one of the slides here as I go through, whether he cringes or not. But it's just a real question of how we go about diagnosing what's not working. So let's start at the beginning. So this was a paper that came out in Nature in 1973 that represents a key beginning. It's not necessarily the beginning of the topic, but it's the beginning of the debate. So this was a paper that Phil Grime published in 1973, and it made two arguments at the same time. That diversity, which he represents as species density, peaks at some intermediate level of productivity in communities. Really environmental stress was what he was focusing on then, and also intensity of management or disturbance. And so he really laid out the if you will, a version of the intermediate disturbance hypothesis before Connell became popular for doing the same thing using a broader array of systems. Immediately after Grime published his paper, there was a rebuttal of his work in Nature, arguing that he was being very selective with the systems he was using to represent this general model. And therefore, one could easily find many exceptions to this model through example. None of this was being done quantitatively. This was all verbal arguments. He responded and countered that, and so we began this process of debating back and forth. And things were quiet for a little while until an empirical paper in 1977 he was involved with produced this graph. And this is a graph that shows along the x-axis the maximum amount of total biomass in the system. And this is herbaceous communities that have substantial component of herbaceous vegetation, and species diversity on the left. And this caused him to take his ideas and to fuse those two notions of disturbance and productivity into a single axis, as we'll see. One of the things that attracted a lot of attention at the time, that's shown up over and over again, is just how selective the use of the data were. So those black dots are from woodlands, and so these are understories of forests. The circles are grasslands. The triangles are tall herbs, if you will. These are really sort of roadside abandoned sorts of sites. And so there's an assembling of data in a selective way to support this. This then was elaborated 2 years later in the first edition of his book into a model which shown down here on the lower right is the so-called humpback model, or Grime's humpback model, in which he argues that there are essentially 5 different processes operating at the same time to regulate diversity. So this was, I think at the time, some really good ideas. There were some interesting theoretical elements that were brought together into this model. And the reaction was relatively swift that people started proposing alternative models with the same prediction. So that was what happened for, a good 12 years after his paper was, well, how about this model to explain the same phenomenon? And the first, chronologically, was Mike Houston's hypothesis of species diversity, in which he essentially combined rate of displacement as a product of productivity. So the more productive communities would have a higher rate of displacement, more disturbed communities would have a higher frequency of reduction, and the diversity is some kind of a three-dimensional model that's controlled by those two. So the real big difference between what he was proposing and what Grime was proposing was an independence, if you will, between disturbance and productivity effects. So it's an alternative expression for understanding, if you will, the same phenomenon. A couple years later, well, this actually 1982, Dave Tillman published a very different explanation for patterns of species diversity that was based on an elaboration of his mathematical model. It shows here a set of equilibria solutions for species coexistence, and you see the circle down in the lower left intersects the coexistence conditions for many species. And as you move out away from the center axis out into more productive environments where resource supply is higher, you get fewer and fewer species that are able to coexist. And so this became his alternative proposal to that to Grime or Houston. And then the discussion began, the debate began about ideas. So there was a series of papers then, 1987 and 88, between Ken Thompson, a collaborator of Phil Grimes, and then Dave Tillman, and back with Thompson and Grime, arguing, wait a minute, you're making these claims, I disagree about who's the better competitor, intensity of competition, all these fundamentals disputed on the theoretical side. So the first attempt to try to consolidate understanding and clear up some of the clearable items was an effort that I put together to try to get people on the same lingo. A lot of the discussion that was going on and was unresolved was because people were using different terms for the same process, or they were using the same term for different processes. A lot of it was semantic and example driven. And so we were able to at least stop some of the debates by getting people to come together, and the book was an offshoot of a series of workshops to get people to talk to each other, to share the slides in their head, to share their perspective. And we got some, for a little while, it was quiet on the theoretical side of things. At the same time that we're doing that, then additional theories are being proposed that are similar and yet different. And so another theory that was being proposed, and this is now the fourth theory that's essentially trying to explain the same phenomenon, argues this what's stated in text here, that richness declines at high productivity because of reduced species pools. And the argument made was that historically in evolution there have not been so many productive environments, and so there hasn't been so much evolution under productive conditions. And so therefore there aren't that many species that are really good at living in high productivity. It's an odd kind of an argument in a certain way, but nonetheless a very different explanation and a different set of implications. So that's four different models. At the same time, additional empirical studies started saying that there were some additional things that were not being accounted for. And this is a study that Laura Goff and myself and another student, Kathy Taylor, did, where we argued that there were environmental filtering of species pools, environmental drivers or controllers of richness all over the place that were having very strong signals independent of production environments. So this was the real key, to say, well, wait a minute, if you look at it to see how much of the environmental signal is going through productivity, you find most of the environmental signal is not going through productivity. And we have evidence to suggest that it's controlling the potential richness, that there are gradients in abiotic factors that represent historical evolutionary filters that influence richness, and those have to be accounted for as well. So just when this is seeming to saturate our number of notions that we can hold at one time in our head, then the other direction began to be discussed when in the mid-1990s, the attention focused to the potential for diversity to have effects, on productivity. So the causal direction being going the other way. And a couple of high-profile papers at that time arguing that higher diversity should promote productivity. Meanwhile, we have all these theories that are based in part on the notion that productivity should lead to a loss of diversity. Eutrophication effects, whether terrestrial or aquatic, all being countered by no diversity should be promoting productivity. So, of course, quite a bit of controversy on this as well followed. Some people saying, well, your experiments are confounded, or it's really just a productive species. It has nothing to do with diversity. There was a lot of early controversy about those promotion effects. And at the same time, all this is going on, the macroecologists working at a very different scale are coming up to the conclusion that the relationship between productivity and diversity at large spatial scales is positive. And a whole set of theories going on there. So now we've got a real complement of alternative possibilities. A lot less attention was paid to the macro studies at this time because the spatial scale, the grain size, we're talking about square kilometers. We're talking about very broad kinds of geographic patterns as opposed to the other studies that were all dealing with much smaller scale units of observation. But as we'll see, these end up plugging together. And there's a whole history here that I'm glossing over that continues to the present. So this first era was one in which there were many discussions about ideas, and a lot of discussions about whose ideas should be considered, and essentially arguing for one case or another. And then the question came up about empirical support. So this is one of my favorite graphs. It comes out of one of Dave Tillman's chapters in the early 90s. But it represents a collection of humpback examples of things. Richness always on the vertical axis, but sometimes biomass, sometimes a stress gradient, biomass litter, various nutrients and so forth. And all of the people who had theories predicting the hump are using the same set of examples in support of their theory. So those of us that didn't have a real stake in the outcome, you're getting rather upset at this point with the selective use of data and no capacity for resolving what was going on. So the era of skepticism really perhaps began with a paper by Peter Abrams, and if you don't know Peter, he's a very well-respected theoretical ecologist, who looked at the theoretical side, looked at the data, and said there's no reason to expect a hump or any particular consistent relationship based on theory alone, and that the data also is consistent in this way. And because he didn't have a theory to compete against the others, then he got a different kind of attention, right? He was like, well, he's trying to pick, and he says none of the above are adequate. The other side of the skepticism began to build in the late 1990s, where people began to look at data, people who were not trying to selectively find support for one theory or another, and concluding that it's really very variable, that you can find as many examples of one thing as another if you go out to simply critique the theory and see if, in fact, it shows up consistently, the patterns that are predicted show up consistently. I did a somewhat similar kind of analysis as well in 1999, in which I concluded that it's a multivariate control that's going on, and we need to consider that in our approach. Kay Gross had another paper that critiqued the consistency, but it was really the paper by Gary Middlebach who really has persisted to this day as kind of a whipping boy for people who don't like these, if you will, naive empirical evaluations and say, well, if we just go and look at the data, we find everything that you could possibly find, and we don't find a strong degree of consistency. So then there was skepticism about the skepticism, right? I mean, you've got theoreticians saying, I can find examples to support my theory, but then you also have more empirical types saying, well, those data are not really very good, or those analyses are kind of biased towards one sort of result or another. They don't have big enough samples, they don't have standardized kinds of measurement techniques, the data are bad. Then that kind of ramped up here much more recently, 2010, when Robert Whittaker, the reincarnation of the original Robert Whittaker, perhaps, I'm not sure, but Robert Whittaker was calling a timeout on meta-analyses as a way of addressing this. The data quality is bad, there's no reason to believe meta-analyses are a profitable way to go, and I have some sympathy for me that he got 1, 2, 3, 4, 5, 6 published rebuttals in a forum. So when he sent this paper and they said, well, let's see if anyone wants to offer a defense of meta-analysis. So at the end, his response to the responses is something rather beat down, like, well, I guess I've had enough critique here, but let me just defend that I still think we need better data, and everyone agreed with that. We need better data that's appropriate to the analysis, if we want to be able to actually say what data say about these relationships. So that was at a time that the Nutrient Network global study was really getting kicked up, and a number of sites around the world, I assume many of you know about this, but I'll assume that some don't. This is a collaborative global study, people participating voluntarily, ultimately to do a common set of experiments, but there was a set of pre-treatment data that was collected with very standardized methodology, and I think with some degree this debate in mind, because the variables collected are ideally suited to this. Peter, were you there at the beginning? Are you one of the progenitors of all this?
Yeah, that was one of our original questions, and we weren't particularly interested in it, but it was the other question, it sort of made it was like, oh sure, let's do this on the way. Let's do this on the way to more interesting stuff.
Funny how things work. They have their life of their own. This is multi-level data, 48 sites with somewhere in the neighborhood of about 40 plots per site, and now I think it's up into the 60 sites or something like that. Different groups come on and add themselves to it, participate, share data, and so forth. So based on this, this smiling young man decided to just say, hey, okay, we've met all of these requirements for data quality. Everything has been standardized. The sampling has been done in a very fair kind of way. And when you look at the data overall, as shown here, and you take all the plots from all the sites and you put essentially live biomass is the measure of annual production and species richness, you see this great big cloud. And if you go into individual sites, you see all kinds of patterns and non-patterns. Rarely do you see a clear hump. And also, if you go to the site level and bin it, the results depend on what you include. So some of the sites that participated were highly altered, like managed pastures where the ground had been tilled and herbicided and then pasture species had been planted. They were very highly altered, and so you could easily get criticized for including those. You can also get criticized for not including them. So the analyses were done at different ways of binning, either at regional scale, across all samples, across samples minus the highly managed ones, and all the results were reported with the strong proclamation that productivity is a poor predictor of plant species richness, which given the data seemed pretty hard to deny. The conclusion was that the signal is weak and variable, and that ecologists should focus on understanding the multivariate links between productivity and richness. Well, I tell you what, it was not taken well. I think these are the hit sequence tips. No! Oh, I'm going to say something about this. This is outrageous. It's wrong for another set of reasons that we haven't discussed here. So, what was really most, to me, revealing and also entertaining in a way, perverse in a way, is that we got these two big criticisms from these two teams of people that simultaneously said, you've done it all wrong. If you do the analyses right, or you approach it right, it's obvious that the relationship is positive, linear, no, I mean, hump. So we got two different groups who look at the same data in the same way and criticize us for completely different sets of reasons. And at first, science wanted us to respond to these independently. Oh, that's not, whoa, no. These answer each other, right? People who want to find evidence to support an idea are looking selectively. And there's nothing inherently bad about that. So when in the process of theory maturation, when people propose ideas, of course, they look to see where they can find evidence that suggests it's important. So, of course, it's selective, and that's the way our brains work, is selective. But eventually, you have to let loose of that and allow data to have more and more of a say. And this is where we haven't gotten to yet, because the selective use or examination of these data was very conspicuous in this case. So our response was, you need to make a distinction between theory confirmation versus theory investigation. Our goal was to critique the theory. Does it seem to be strong enough, the collection of predictions strong enough to continue using this relationship as a basis for justification? Well, okay, that really got things wound up. We didn't settle anything with anybody on one side or the other on that one. So then there was a paper that came out earlier this year arguing not only that our analysis was flawed and there was a very conspicuous hump if you just selected the data across the top of the point. So if you go to the top, you just pick the points across the top, and then you do a curve fitting exercise. You can explain, at the site level, 98% of the variation of the upper boundary, made up of 7 points. And it's important that we do that because of management. How are managers going to know what to do if we don't make this the basis for management? It's like, you're only using 5% of the data. So what do you tell them about all the other places? So anyway, this one we had no capacity to avoid. But another debate came out, and a couple things really helpful happened. So at this point, this collection of four, there actually are five, only four loud voices in this, that are all predicting the same thing, but they begin to separate themselves. Okay. So here, this paper is now only talking about Brian's compact model. Usually only talking about his dynamic equilibrium model. So that makes our job a lot easier, because we're not now talking to a disparate group of people who have very different terms and drivers and so forth. There's also been a call to do it right. So there's a paper about a forthcoming paper. It says, well, like NutNet, we need to get everybody together, but unlike NutNet, we're going to measure two variables, standing biomass and emissions, but include a broader span. And including broader span, that's great and that's fine, but just trying to make the point that we really didn't resolve things. But what we did do is to take a step back and say, look, our point is that we can demonstrate on many different levels that bivariate data plots are not adequate to separate 4 or 5 different theories that contain a collection of, say, 20 different processes. There's something called identification. You know, identification is when you take a class in linear algebra and you have to have 2 equations to get unique results for 2 unknowns. If you have a lot of things at play, you have to have an equal amount of information. So looking at a bivariate plot, you can come up with many competing explanations, and you can never resolve which ones would be operating, even if you had a strong curve. Well, it hasn't slowed down the folks who are disagreeing with our perspective, but maybe we're at least giving them some new things to think about. So here we are, 40 years later, and I could say that with the exception of the one effort that I've led, I've not seen any attempts to reconcile or consolidate or make progress with this. So this is one of those cases you hear about where wiring gets tampered with and not re-engineered, but just added onto, and I think this is a collection of ideas that we have. There are a couple different summaries that have been done, counting up all the different theories of diversity and so forth, and there are many, many out there beyond these, and we don't have any, they're accumulating. They're not being falsified, they're not being validated, they're just accumulating as additional explanation. So where are we going with all this? Well, I'm going to argue that maybe there's something else that we can do. There might be another approach that we can use. We haven't actually, I didn't turn off the front set of lights here. Can people see okay in the back? These all showing up? Okay, that's good, because I don't know for sure which button I was supposed to push. So it's probably best I leave it alone if I can. But the question I'm trying to get at here is, what else can we do? Well, there is another approach, and that is to conceptualize systems in terms of probabilistic causal networks. And the argument that's being made is that probabilistic causal networks are an inherently effective way of summarizing information, reasoning, and developing testable hypotheses about systems. This graph comes from the cover of a book that's the progenitor for modern artificial intelligence. And so the folks dealing with AI have grappled with what should be our fundamental framework for assimilating information, updating information, making forecasts, reasoning forwards and backwards. And their conclusion is that it's a probabilistic causal network. And so there's another way we can approach systems, aside from trying to find consistent bivariate relationships or even single models with 3 or 4 processes. So let me say a little bit about this enterprise of causal networks and the associated structural equation model. So structural equation modeling is the methods that we use for building and evaluating hypotheses about systems in that form. And it represents a framework for using statistical and mathematical tools to evaluate hypotheses and learn about how things work in systems. So it's not a particular statistical technique, it's really a set of principles that form a framework. It's also a way of translating ideas. And so here I show what we did in that functional ecology paper that we published this year, where we took Grimes' model and we said, well, if that's your model, here are the testable implications of that model. So you say that stress up there has an impact on drives productivity, which in turn shows up as effects on biomass that controls maximum potential species richness. we have to assume that there's some relationship with observed richness to maximum potential richness, which you say are influenced by species supply and its differentiation, and the disturbance comes in here. So this is literally what you're saying. This is testable. We can test this with data. We did test it with data. I won't present those results now. And what we showed was evidence for these processes and a whole bunch of other processes much stronger than those processes that come from data. So I'm expecting a paper to come back saying, that's not what I meant. But hopefully it's in this framework where we can now talk about it without just never getting a handle on what can be evaluated. It'd be interesting to see. I can't imagine that Phil Grime will not come back to something. It's not possible. Another thing about structural equation modeling is it has a particular form. So for folks who have a background in statistics or some exposure to that, we have the equation y is some function of x and some vector of x's plus some unspecified set of factors. And in structural equation modeling, we have this extra term that I've outlined, that y's, response variables, are also a function of other y's. And in the non-parametric form, we say that y's are functions of x's and y's, plus the things that are omitted, the unspecified. And by allowing y to be the equation, now we can represent networks. And I think it's, I'm not alone in feeling like that the network structure of a hypothesis is what you really need if you want to be able to investigate and falsify causal hypotheses, test ideas about mediation, find places where things are going on that you're not expecting. There's something special about this network that has big advantages. It's also, SE is also a method of learning where you might start with some kind of relationship which has a net effect. This is what you get out of classical statistical model, is some kind of net effect. But then you hypothesize an explanation for that net effect, and then you can test whether that seems to be adequate, partially adequate, or completely inadequate. And so it's a method whereby we examine the variables that are in our model and say, well, why is that in the model? And can I prove that the proposed explanation is adequate? And if not, then I find something else. And when we find something else, this represents a new process, a new way that, in this case, the A to the fourth before it burns can influence plant recovery independent of fire severity. And so this test of mediation is a critical feature that comes as a way of learning about systems through the evaluation of network hypotheses. And then also is a process. SEM represents a process of translating theory and models, obtaining measures, evaluating competing models, getting adequate models eventually through a comparative diagnostic process, and then revisiting the implications for theory. So those are just some of the main characteristics. But I think the history is very interesting and important. So the origin really goes back primarily to Sewell Wright, not exclusively, but primarily to Sewell Wright. Sewell Wright was a biologist, a geneticist. That whole early cadre of Fisher, Wright, Galton, and Pearson, they were all really human geneticists. They were really interested in the genetics of humans. They were very interested in human populations. And Sewell Wright was famous for working on guinea pigs. And so guinea pigs were his primary subject. He would look at correlations amongst bones or breeding results and so forth. And so here he is lecturing with a chalk in his right hand and a guinea pig in the left hand. Some people are catching on here. So the rumor has it, and if it's not true, I don't want to know it, that he would occasionally turn around and erase the board and go on. Some people have said that's not true, but like I said, and so in his, starting with his master's thesis, but then in his PhD, he developed graphical modeling and path analysis all at the same time. So the whole concept of using graphs to represent causal logic goes back to him, and then also path analysis and what has become causal modeling dates back to him as well. So that was the predominant approach until the early 70s, when Carl Jorskog, shown here, with a very different expression on his face, did a major synthesis of mathematics and produced a set of equations that were much more general and solve a much broader set of problems. that we can imagine that there are some set of latent variables that lie beneath the observations that we see. That we see manifestations when we collect data. Causes are invisible because they're happening. And so we represent the causal variables as latent variables, and we draw inference about what they're doing from the patterns of observation. This idea goes back to the attempts to understand human intelligence by asking sets of correlated questions, right? So you test individuals and you give them a whole set of questions, and you look to see if some people consistently get higher scores on all the different categories, or maybe they only do higher on verbal and not on math, or maybe analytical is different, or you start to look at the responses and draw some understanding about what the underlying causal process is. So he developed an integrated set of mathematics that summarizes all those equations into a general formulation of latent relations and measurement relations. I like to say the reason he's smiling is because when he developed the mathematics, he also developed the software to run the mathematics. So Sewell Wright's there going doing everything by hand, right? And Carl Jorscott is going, hey, this is great, man, I can check my internet, my email. So really a brilliant advance. There's some interesting history there as well, because there was so much interest at that time in being able to fuse the fields of econometrics and psychometrics and biometrics into one system that could do them all. They held a competition, and they got about 8 different mathematical theoreticians, statistical theoreticians around the world, and there's this book in which each one of them attempts to solve the problem. And the clear winner in all that was Carl Jorscott, who I think, in part because of the practical nature of his implementation, his equations and his implementation all came together in a way that was quite revolutionary at the time. That's when the name changed from path analysis to structural equation model. And biologists a lot of people didn't pick up on that name change at the time. The inclusion of latent variables, by the way, allows for a lot of additional kinds of models to be evaluated. So here we are looking at floral resources in New York City in relationship to flower visiting insects, and our theory is inside the box. It's among the latent variables. And everything out here are the observed manifestations that we're using to draw inferences about that. And the one additional thing that came over in this from econometrics was the ability to start asking questions about causal loops, or evaluating models that have loops that go around and have causal feedbacks. So that was a primary concern of people in economics, because that was the first question that quantitative economists dealt with. How do we deal with simultaneously interacting variables like supply and demand? And this formulation allows those models to be estimated. So I think one of the more exciting, very recent developments has been the fact that the field of AI has embraced structural equations as the fundamental language of causal reasoning, and also that AI needs to depend on causal reasoning. So this has kept me very busy, because a lot of new information has come out. The first generation of AI, based off of Pearl's book on probabilistic reasoning, led to him getting the Franklin Award. He then followed that up with his book on causality, once he decided that if we don't use causal reasoning, and we just use associative reasoning, we're never going to get to a functional AI. So therefore, he went back and actually rediscovered Sewell Wright's work, and then has redescribed that work, which now led to him getting the Turing Award, the Nobel Prize in Computer Science a couple years ago. So I like to show this graph because, now can you see this in the back? Okay, all right. I don't have to take a chance with the lines. It's probably good. So this is a science map, a historical science map that shows how the sciences are connected based on cross-citation of fields. And I think it explains, potentially, explains a lot of things. So ecology and evolution, we're over here kind of on it. Well, everybody's, I guess, almost everybody's on some kind of edge. So it does seem that we're kind of separated from everybody else, but I suppose you could say the same thing if you were a geoscientist. medicine, whatever. But we're over here, very close to crop science. Now, that doesn't surprise me at all because the other dominant figure that actually predated Sewall Wright by a few years was Ronald Fisher. And Fisher's first job was at the Rotherham State Experiment Station, which was a big ongoing experiment. So he focused on experimentation and agricultural kinds of settings. And Fisher was absolutely the 800-pound gorilla in the world of statistics. Very, very smart, very aggressive. People still go back and look at the things that he said about other people. He pretty much destroyed Carl Pearson at a personal level. So he had a huge, he had a huge influence, and it's over here. And so biology has been very closely aligned to the statistics that is translated through agriculture. Meanwhile, on the opposite end of the world, we have economics. Economics has always been system-oriented. It's never been a reductionist kind of a science because you can't really do experiments on economies very easily. So they've tended to adopt actually, Sewall Wright's ideas came over here and have led to human sciences because of a predisposition towards systems kinds of ideas. And so structural equation modeling, we've been largely living over here the whole time. Probability statistics, by the way, up there, and up there, computer science is where AI is. So I think this explains a lot about the disconnects and different terminology in the literature, different perspectives and so forth that exist. So for the past few years, myself and a number of folks have been working to try to pull SEM to a more central location, to try to strip it down or deconstruct it, if you will, to pull out the essentials and separate them from the measurement issues that preoccupy people in human sciences. And so that's been part of the effort. The other part has been to try to integrate these ideas of AI with modern statistical methods like Bayesian analysis, and also the kinds of concerns that we have in ecology, all into a third generation SEM, which we published a paper outlining that a couple of years ago. So, how might this help us with regard to productivity and diversity? SEM begins with a translation of ideas. And so here we have a word map, right? It just pulls common words out, and you see all these words that represent the ideas that are embedded in this debate: richness, diversity, biomass, productivity, heterogeneity, etc. So what are the key theoretical constructs? I'm going to unfold this a piece at a time. Hopefully it won't be overwhelming, but it's the best I can do. So the dotted lines mean that these are defined linguistically. And the precise way that you might measure them, whether it's one variable or two or a particular measure, is still unspecified, undecided at this point. But I think that there's some basics that are pretty easy to justify based on what we know. That productivity is connected to diversity through a sequence of events. Productivity leads to biomass accumulation. Biomass leads to resource reduction, which leads to impacts on diversity, and diversity can feed back on productivity. So there's a part of the debate that's been out there about what should I be measuring? Should I be measuring biomass or production? Is this adequate or that adequate? Well, this shows that there's a logical connection between those two things. They're in the same causal chain, but they have distinctly different properties to them. Another part of this is disturbance. And so disturbance is a key concept, and it's part of the reason that biomass is different from production. It's not the only reason, but it's part of the reason that biomass gets produced, and sometimes it gets consumed, and sometimes it doesn't get consumed. So disturbance is part of the difference vector, and disturbance can also influence diversity as well. The bottom-up part of this is resource supply. This is another thing. If you think back to that graph with all those different humps, and it said some of them have nutrients, some of them have biomass, some have biomass plus litter. Resource supply is also in that causal chain. This one is one that I think people may have the most trouble wrestling with. My argument is that the abiotic milieu in which organisms live functions quite independently on diversity than it does on productivity. We have a lot of empirical evidence to show that when we look at the, say, soil conditions or chemical conditions that should be driving productivity in the colonization of species, that these two things are very uncorrelated, even when they're made up of the same variables. And so I think it helps us at this point to say that those environmental factors that are affecting diversity, independent of productivity, we're going to call environmental filters at this point. And that term is becoming increasingly used in a number of different fields. So climate is another big driver. We have to understand that these big gradients that produce large species pools, if you will, in different areas, high productivity areas having more species than lower productivity areas, can filter down to our sample. And so there will be effects both on productivity, but also effects on diversity. And heterogeneity is the one other factor. So this is where we haven't talked much about Dave Tillman's theory. But Dave Tillman's theory is really one that depends on heterogeneity. Now, there are many other heterogeneity explanations, but the amount of diversity in an area, for many reasons, is seen as being dependent on the heterogeneity within that area. And that more monotonic areas won't have as much niche space compared to more heterogeneous areas. There's a body of debate about that as well, but maybe less contentious. So, as I've sorted through to try to find the minimum number of constructs it would take to distinguish the different theories, we've come up with 9. Sorry, it would have been really nice if it was 4, but it comes up to be 9. And the question is, does anybody have the data that it would take to evaluate this? Couldn't resist putting a long range right there. 39 of the sites in that have measured an adequate set of variables to be able to represent those 9 constructs simultaneously measured in the study. So this is something that we're working on. The model that we can develop includes measures of resource supply, productivity, climate, disturbance, shading as our resource reduction variable, and for diversity, species richness. For a variety of reasons, species richness has been the variable that everybody's kind of locked onto, even though we talk about diversity, and sometimes we do look at functional diversity. But these things are all represented, and here the drawing is meant to give you the mental context of the evaluation. These are herbaceous communities. Seed bank is very important in regulating them. Shading has an obvious kind of effect on suppression of seedlings and so forth. So the context for interpreting these concepts is perhaps improved, even if the range of generalization is narrowed a little bit. So what do these bivariate data suggest we will find? So here are our plots, color-coded by site, plotted on a log-log basis because the variables are highly skewed, and the signal is on a log transform basis. So this is what we're looking at, and I think it's I'm going to argue that it's kind of like one of those accordion dolls that when it's compressed, it's just this sort of amorphous thing. So the question is, can we pull it apart and see the component pieces with the model that we just proposed? So, unfolding the results here in sections from left to right, we see that productivity at the site level, so these are site level results, strongly related to resource supply, particularly soil phosphorus is a key driver, and climate, particularly precipitation and temperature. Biomass, very much determined by productivity, slightly offset by the disturbance, and particularly grazing in this case. But this particular set of examples, I think, is a low disturbance set of examples, at least contemporaneously. Historically, maybe not. And then richness, influenced by a whole combination of things. The first thing I want to point you to is that I bypassed shading because our one-time shading measure doesn't seem to be adequate to conclude that shading is the only reason that biomass has a negative effect on richness. Climate effect on richness, independent of productivity, very strong. Species with historic human impacts are lower in diversity. Heterogeneity, the more heterogeneity, the more diversity, so that was actually a pretty strong effect. And strong environmental filters. So this is the sort of the first set of signals that show up in this model. The other, which is one of the key things that we were looking at, is could we pick up an effect back from richness on productivity? Because when you look at that cloud of points, there's no way you can pull a positive relationship out of it by itself. Most of these things. But this is one that has evaded us in natural systems to be able to pull this simultaneous negative effect of productivity, i.e. competitive effects presumably, separate that from positive feedback from richness to productivity. So it shows up at the site level quite strongly, actually. This is what surprised me. So here are the key relationships. So once we start doing the partials, we control for the various things, we see this is what the shape of the biomass effect, if you will, the competitive effect is. It's strong, it's reasonably linear looking. At the same time, the productivity effect is also strong and positive, and then that macroecological signal from precipitation onto richness also sorts out. So we're pulling these things all out of that cloud of points in bivariate space. So it's about the message here is about the dimensionality of the model. We need a model that has enough parameters in it that we can begin to disentangle all these things that are happening simultaneously at this scale. So I have done some analyses to say, well, how many things can I pull out of this and still be able to recover the clear signals that emerge? And the answer is not much. These three variables here have some reducibility. You can pull out biomass and the productivity come through, you can pull out resource reduction, but anything else you take out, you start losing paths, and the relationships stop becoming linear and, if you will, parsed. So a few pieces can be removed. So I want to say a little bit about scale, because scale is one of the things that people talk a lot about, scale of observation. Here we are on the planet. There are global scale studies, regional scale studies, smaller scale studies, and then single point kinds of studies at least at this scale. And we do have data within each site. So within each of these sites, we have replica plots. And so we can ask, what about within plots? So these are essentially the deviations from the mean in productivity. You see that biomass is really mostly predicted by the site level biomass. But at this scale, shading does show a strong relationship to biomass and is an adequate explanation of the effect of biomass. So at the small scale, our one-time measurement of light seems to provide a fairly clear explanation for that effect. And we do still pick up a reciprocal effect. It's been disputed at this scale, although it is quite a bit weaker. So here are the key relationships that tease out at the plot scale. So this is shading with biomass, the loss of species with increased shading, and then this is that small-scale biomass, or the productivity enhancement, if you will, due to richness. It's really quite marginal, if you will. Some people will not like this, but that's what you can. So we can combine these two together, and by combining them together, I know this is quite messy, but it does allow an integration of macroecology with site level, plot level, drivers at both scales into a system. And the question then becomes, what are the fundamental attributes of this system? So in sum, we can say that behind that cloud of points are a whole bunch of clear relationships when you have a model that includes enough of the factors in it. So the particular things that show up that I'll mention is that these positive feedbacks are detectable at both scales, and that was very questionable. The other is that competitive regulation of diversity has a clear signal, at least what we're interpreting as that. And that species pools interact with environmental filters. That they're behind the scenes, there's this variation in species pools driven by environmental filters that have an independent effect that's quite separate from these other processes. So what would a resolution to the great debate look like? I don't know how many people are now familiar with Murder on the Orient Express. Do you have any? So the movie Murder on the Orient Express is a classic Hercule Poirot, Agatha Christie novel in which a murder takes place on a train, the Orient Express. There's a lot of investigation that goes on, and in the end, it turns out everybody did it. Everybody played a role in the murder, but the roles were not all the same. Some were very minor, some were very major, some people actually applied the poison, other people were just distracted. And so I think that any resolution of this set of relationships between productivity and diversity has to account for the fact that there are several things at least going on, but the question becomes about how they interact together and the relative importances. Many processes play a role, but they're not all equally important. And that overly simplistic models hide the signal, or alternatively, they would bias the signal by attributing everything you find here, say in a hump-shaped fit through this data, to one cause by ignoring the other causes from the model. If you have any interest in learning more about these methods, we're just days away. Well, hopefully, by the end of this month, we'll have a training website where we're going to be giving more methodology and more exposition. So, thanks for hanging in with me. I know this is a lot to cover, but it's kind of the nature of this story. It's a complicated story. Thank you very much.
So, I think partly because of the technical troubles at the start, we're up against 7 o'clock. You have to leave. We'll stay for some questions. Folks who are leaving now, don't forget to come to the reception at Zach's Conference House. Address up there, 77 North. It's all fun. Questions?
Well, that's usually a sign that you've been
You mentioned if you take a model
right.
where gas actions, like I would assume like accidents
So one of the points I tried to make along the way is that these models are sort of self-purifying. In the sense that you run the model and test it against all the alternative models with the different kinds in there. So there's a lot of reasons you might imagine additional mechanisms beyond it. The data don't suggest we need to miss it.
All the relationships like linear.
So the assumption of linearity is a great question, as well as other assumptions. In this model, we are modeling things as a linear approximation. We're partly doing that because the complexity of this defect forces us to use more generalized techniques. We can do polynomial fits, so we can do curve linear fits using polynomial terms, and we do that quite often. So it is possible to do some curve fitting on things that are not strictly linear. There's no real indication. If you look at those scatter plots of data versus prediction, I was quite surprised that they were, quite surprised, I was shocked that they weren't as good as that. It was really quite honest. Yeah. And when they aren't linear, the question is a linear approximation with most of them? Because things can be pretty clearly not linear. If you have something that saturates, you fit a straight line through it versus a curved line, variance explanation only goes up less than you might think.
One more.
You focused on the plant aspect of things. Has there been much debate in other systems on this topic?
You know, there has been far less debate about the animal side of it. There has been some. There are a couple, particularly in the macroecological realm, where it's pretty much just as easy to ask why there are gradients in animal diversity as plant diversity. But it's mostly been amongst the plant people. And so the model is constrained by the fact that I'm just trying to figure out the debate between these two variables. It's not in any way a complete model. Thank goodness for that. Wouldn't want to be there for that talk, right? Good question.
So Jim, wouldn't an exception to that be food web analysis we've seen structural equation models applied in? I mean...
Oh, you can use structural equation modeling in all kinds of cases. In this case, this model is designed to try and shed some light on this particular debate. But Yeah, the structural equation modeling can be used on food web analysis. I taught a workshop in Germany in February where the first 2 days were all network analysis. The last 3 days were how to use SEM on those data. So yeah, SEM is very versatile. This particular application is customized to a particular problem.
Do you have any closing comments on what R packages you use for SEM?
Well, so in this case, I use Levan. So Levan is an R package that's kind of the up and coming. By far the best, most powerful package is N+. It really is very powerful, but it's expensive. I teach all these workshops. I can't ask people to come $1,400 to come attend the class. So we're hoping that Levan can get enough contributors. They're trying to catch up with N+. Amos is another good one for Entree, but again, it's a commercial software package. And so we're going with R for sort of obvious reasons, and Levan is the best one we do right now. There's another package called SimTools that has tools for querying or working with Levan objects. There's SimPlot that you can run Levan objects through there to get a graph. There's a Levan Survey, which I used in this case to deal with the nested nature of the data. So you tell it that these are nested, plots are nested within site, and it adjusts the confidence intervals based on those structures. And so it's progressing pretty well. So on that website I talked about, I'm laying everything out initially in Levan as the examples, with then some other implementations.
Okay, let's continue the conversation at reception.